Good morning, everyone. Thanks for coming. We talk about the crystallography facility at the LMB. I am managing the facility under supervision of David. We share our room with Fabrice and the crystallization facility, which is under the supervision of Jan. So Fabrice already showed you in a previous lecture everything about product crystallization, optimization. So the question is, once you have the crystals, what do we do? We're going to put it in an X-ray beam. We're going to get a diffraction image. We're going to get many diffraction images over different oscillation or angles of the crystals. And then we can convert these ones into a real space electron density map, inside of which we can build our model. So just very quickly, one crystallization trick, which is a personal favorite of mine. If you're struggling to get crystals, what you could do, you could add a protease, try to cleave off your loops. Do not run a gel, because if you run a gel, it's going to look like a mess. But in reality, the protein is still intact. So how the protocol works, we add the protease. Hopefully, we cleave off your flexible loops. Then we add an inhibitor. And then we put everything into crystallization. Hopefully, we're going to get crystals. We got 3 different proteases available at the facility. So if you would like to try with some of your projects, please let me know. This is an example how we helped one of the students here before. This was the crystal we were able to obtain. And this is the structure we solved, which went in a good publication. To begin, the hits were like this. And then just through a couple of rounds of optimization, we got suitably diffracting crystals. Few things you need to know. Once you get crystals, you cannot just freeze them. Well, sometimes you can, depends on the crystallization condition, but usually you have to apply a cryoprotectant, which will stop the ice from the water to become ice. So we keep it like a form of vitreous ice. We can use the same procedure to add the ligand into your crystals. For example, if you're doing some drug discoveries. Difficult in soaking depending on your crystals, really. If your crystal is very robust to osmotic shock, then it's going to be easy. If your ligand is very soluble, so you can use high concentration of it, it's very easy. This heavy atom, so also heavy atom needs to be soaked in, but this is becoming less popular now that there is AlphaFold. I will show you later. So the different level of expertise are required, but if you've never done it, don't worry, we are there to help you. Once you grow the crystals, you will need to harvest them from inside the drop. These are some of the tools that we are using at the facility to harvest your crystals. So this is your crystal in the drops. These are the tools we use to fish them out and freeze them. And once you freeze your crystal, they can also be shipped to synchrotrons. They will be shipped inside these containers. That's how you use a magnetic wand to place the loops inside the pack. Once this pack is full, you're going to close it. Of course, all of this is done under liquid nitrogen. And this will be shipped in a cryo viewer to the synchrotrons. However, we do have an X-ray generator in-house, which uses a copper rotating anode. And we have a 2-station. So 2 people can work independently. We got 2 image plate detectors. And again, if you need training, you need access, you need help with it, you can just come and see me. So I'll take advantage of our X-ray generator in-house. We can quickly screen your crystals. We also have a cryo oil, which means you need to make a cryoprotectant for all your different condition of crystals. This is like a universal one, which is good enough just to screen the crystals, which will quickly tell you whether it's a salt crystal or it's a protein crystal. In case it's a salt crystal, you will know that you don't need to invest any more time to optimize this crystal. You can move on to your next hit. As I say, you can use to optimize cryoprotection. That will help you to minimize the time that you spend at the synchrotrons, because sometimes you can get like overnight hours. Instead, here you can work during the day. Sometimes the synchrotron is closed for maintenance, so actually there is no access. I mean, you can collect it 2 to 10 minutes per image, so we usually do it overnight. Also, we can select your best crystals. For example, you need to use like a synchrotron beam like I-23, where they do not have high throughput. So you can only ship to them like 5 or 10 samples. You will need to find your best one before ship them. Okay, if the beam line is closed, if the diamond is closed, there is no beam time. So the synchrotron we are using is the diamond synchrotron, which is going to vary a selection of different macromolecular crystallography beam lines. Depends on which experiment or the type of your crystal. We can select the right one for you. Again, to request time, just drop me an email and we can arrange everything. To data collection option of the synchrotrons, we can either go there by in person, which I think is a good experience to visit how the synchrotron works, how the beam lines are built. Or we can just, as you say, we can just ship our crystal in our packs, and then we can collect remotely from the LMB, from the X-ray facility. There is a nice station with 2 screens. So data collection strategies, a bit of theory of crystals first. So inside the product crystals, what you have is a crystalline arrangement, which means that the crystalline arrangement is defined by a lattice, which is an infinite array of points with identical environment. Now, if you associate a molecule with every lattice point in the same orientation, that's when you get the crystal. Within the crystal, you can find you can always find the unit cell, which is the smallest basic building. That just by repeating it, you can rebuild the whole crystal. Like these are all unit cells, okay? That's what we mean with unit cells. These unit cells, these lattices, they can only have 14 types, which are divided in 7 systems and 4 types: primitive, base-centered, body-centered, and face-centered. And the system are the triclinic, monoclinic, orthorhombic, trigonal, tetragonal, hexagonal, and cubic. However, within the unit cell, you can always find something we call a symmetric unit. This is the smallest unit that can reconstruct the whole crystal, but you need to apply some crystal symmetry. While the unit cell, you just need to translate it, X and Y, to reconstruct the crystals. As you can see, here we have 2 identical asymmetric units within the unit cells. But as you see, that they are rotated by 180 degrees. So we said that there's a 2-fold axis between these 2 that relates these 2 asymmetric units. And this will come, I will explain why it's useful later. This is a real example. That's how you look your protein crystals when you solve the structure. So you need to become familiar with the word space groups. You will hear this a lot in crystallography, and the space group of a crystal is just a concise description of all the symmetry in the given 3-dimensional crystals. Crystal symmetry come in different flavors. You can have rotations. You can only have 2, 3, 4, or 6 rotations in crystals. You can have inversions, reflections, screw axes, and glide planes. So let's see, for example, if I say to you, this crystal has a space group of P4122. What it means, the P means is the type of Bravais lattice. If you remember before, is the primitive type. The larger number, 422, are the rotations. This is rotation along the C edge of the cell, and the 22 are along the A and B sides of the unit cell. The smaller number instead represent the screw axis. So there is also a screw along the 4-fold rotation axis along the C edge. That said, there is a total possible number of 230 possible space groups applying all the symmetries together. But that said, because the amino acid, the building block of the proteins, are chiral in nature, this prevents some of the symmetry to exist. So you will never find inversion, reflection, glide planes in protein crystals. Therefore, the combination of screw axis and rotations only allows for 65 space groups to exist in the protein world. And these are all the 65 space groups that you will ever encounter in protein. A little caveat, there can be some very special cases where you might find the 5-fold symmetry or the strange things, but these are super rare cases. So data collection strategies. In-house, as we said before, it takes 2 to 10 minutes per image. At the synchrotron, it takes like tenths of a second per image. So we do larger oscillation in house. So the way you collect the crystal, you oscillate it by 1 degree. You will need to estimate your Bravais lattice. At the synchrotron, you don't care because it's so fast that you need to do any calculation. You want to estimate this so you will know how many images you need to collect. Obviously, if you have like a high symmetry crystals, you only need to collect 30 images, which might take you only 1 hour. Instead, if you have a low symmetry, you need to collect 180 degrees. So that will take 5, 4, 6 hours. So you need to take into account these differences because there's a big difference whether your experiment needs to run for 1 hour or 6 hours. 1 hour, you can do like about 10 crystals in a day. 6 hours, you know you're only going to do 2 crystals in a day. You don't care about this at the synchrotron because whether it's 30 degrees or 680 degrees, it's still going to take seconds. Mosaicity, this is what Fabrice explained in his talk. So the problem with mosaicity is that because you are doing large oscillations, you might have overlaps of spots in your detector. That's something is not going to happen in the synchrotron because you do very fine slicing. However, the problem that we don't have in the house is radiation damage because the beam is so weak. But this is something you always need to be aware at the synchrotron. You can check this on the fly. This is one of the tools. For example, you can see the red dots showing the resolution of your diffraction image starting to degrade. So you know that you are hitting radiation damage at this point. And as I said, this is the data collection is critical. If you spoil your crystal with radiation damage, you will not be able to recover it in data processing. You have to go back to the synchrotron and collect a new data set. So be very careful when you choose your studies for data collections. So how do we process then the images we collect? Let's have first a quick look at diffraction theory. So imagine we have like out of the whole crystal, we take only a single unicell, okay? Like a single molecule. We hit with an X-ray, the molecule is going to scatter your X-rays, and this is what you see on the detector. However, because X-rays scatter with atoms like carbon, oxygen, very, very little, it's impossible to detect the scattering from a single molecule. That's why we need to use the crystals to amplify the signal. How that works? Because of the nature of the crystal of billions of unit cells, the signal gets amplified over a reciprocal lattice. But the payoff of this diffraction is that you're only going to see scattering or diffraction where the lattice points are. And so the image you're going to collect with your detector is what is called a weighted reciprocal lattice. You can see that there is underneath, there is the image, but it's only seen where the reciprocal lattice points are. This might not look very familiar to you, but this, if I show you something like this, now you're going to recognize the detector image of X-ray diffraction. And this is what you see on your detector, is the weighted reciprocal lattice. So that's how they used to teach me crystallography in my university lecture. I don't know if this means anything to you. It didn't used to mean anything to me. So I went to dig a bit deeper. I'm going to try to show to you how understanding these things works. So each spot you see here, they'll be called structure factors. Each of them will have an amplitude, a phase, and 3 numbers. And these numbers are not random. We're going to look now why we're going to get an amplitude, a phase, and these 3 numbers. These 3 numbers are called the Miller indices, and they are H, K, L, and they are related to the edges of the cell. H is related to the A side of the cell, K is related to the B side of the cell, and L is related to the C side of the cell. How we are able to predict, how we are able to turn this into the real electron density of a protein is thanks to Bragg's law, which you might have seen before. It's very beautiful and very simple. 2D sine theta equal lambda. Well, what Bragg's laws basically predicts the direction of constructing interference of scattering waves. And now we're going to see what this means, okay? So imagine you have a wave, an X-ray wave, of wavelength lambda, okay? So these are all your equal wavelengths. And now you sit in the atom A. At this point, your atom will start to scatter the X-ray in all directions, okay? Now let's consider if you have another atom at the distance D, okay, that's our D, the distance between two atoms. And we have our coherent, of course, the X-ray beams needs to be coherent, parallel coherent, which means all the beams, all the waves needs to be in phase with each other, okay? As you can see, these are all in phase with each other. Now let's say now B is sitting by node, and it's also scattered in all directions. What Bragg's law is telling us is that there is only one theta angle of the scattering radiation where the two waves coming out of the atoms will be in phase with each other, okay? So for this to be in phase, it means that this path needs to be half wavelength on each side, okay? Or one full wavelength, or 1.5 wavelengths. We will see later in a second. I can preview this quickly like this. If you construct it like if you do a geometric construction over a different direction, you can see you are not in phase anymore. There will only be one direction where this is in phase. Imagine the scattering angle is your 2 theta angle. Now imagine we draw an imaginary reflecting plane or mirror plane that acts like a reflecting plane. Okay, the theta angle here is identical to the theta angle on the other side. Okay, these are just imaginary planes, just to help you visualize. Bragg's law says that every atom, A or B, that is lying on this plane, it will always scatter in phase with one another, okay? And what it turns out, this direction of to satisfy the Bragg's law, if you have like a fixed lambda, okay, it happens that if this direction D decreases, gets smaller, this angle becomes bigger. So your plane is kind of, your mirror planes are kind of rotating this way, okay? Your reflecting angle becomes higher. If this distance between these two angles becomes larger, you see the reflecting angle becomes shallower. So the waves scattered in phase with each other will occur at a lower angle. And this, so smaller d equal larger signs. So we are talking that the information at lower resolution, because let's say these points are like 5 angstroms from one another, okay? And these points are only 1 angstrom from one another. So the information about the points that are 5 angstroms from each other, which means the low resolution information, will be scattered near the center of the detector. Instead, the information carrying the high resolution information will be scattered away from the center of the detector at higher angles, okay? You will hear this concept a lot in all sorts of crystal, even in cryo-EM and also of structural biology. As I said before, any atom, let's say even A, B, C, or D, that is sitting on these planes, it will always be in phase with each other. Okay, so it will always scatter in phase for the angle that satisfies the Bragg law, which will be in phase with each other. But how about if there is atoms? There could be atoms in between the planes. Okay, do they not diffract? Do they not scatter? Of course, they scatter as well. But if you notice here, for this one to be in phase, okay, it went from one wavelength. There's an extra wavelength, okay. So we went from 0 to 360 or 2 pi in radians, which means back to 0, okay, and back to 0 again. That's why everything on the plane is always in phase, because they go a full cycle, and go back to 0, okay. Let's say, for example, you happen to have another atom who was exactly in between the two planes. Its phase would be 180 degrees, okay? Will be only half wavelength away from the other wavelength. So which means now we'll be completely out of phase. If you had another atom that, so intuitively, if another atom was sitting here, like a quarter of the way between these planes, its phase will be about 90 degrees, okay? So how can we use this information? Let's see what happens in a real crystals. You got real crystals now. you got your parallel coherent beam coming through, and there will be an angle for which D will be a given D that will randomly cut through the unit cells, okay? So now you can see this atom here in this unit cells will have 0 or 360 phase, okay? But this one is not sitting on the plane, so it's not going to be 0, it's going to be something else. This one's going to be something else. This is going to be something different, which means over the whole billions of unit cells, the signal will get cancelled out. Instead, if you find the right direction, where your plane are going to divide your unit cell by an integral number, what happens that the same atom from each unit cell is going to be with the same phase, okay? 0, 0, 0, 0. Let's say this is sulfur, for example, it's going to be about 90 degrees, 90 degrees, 90 degrees, 90 degrees. Which means now, whatever you get out from one unit cells is going to be amplified over the billions of unit cells. And that's where the HKL comes from. If the family of planes is going to divide the A edge only once, you're going to get 1. If the B edge is going to be divided by 4 times, you're going to get 4. If you consider now that the C edge is coming out of the screen, our planes are parallel to the C, they will never meet the C edge, you're going to get a 0, okay? Now, as we said, we can have many of these planes, these families, okay? For example, this is the HKL210. But we know that the larger the distance, the shallower the theta angle. So if this beam is coming here, if this is acting as a reflecting plane, the beam should come up about this direction, okay? Which means it's a very high theta angle, which is not going to satisfy our Bragg law. So what it means, if you want to catch the diffraction from these planes on our detector, we need to rotate now our crystal, okay? We need to rotate our crystal so that our plane now can reflect the beam at a shallower angle, which satisfies Bragg law and can be seen also in our detector. Our angle, this angle needs to be, it can be easily calculated using Bragg law. If let's say our D plane is 10 angstrom, our lambda is 1 angstrom, we just rearrange our Bragg law equation, and we can find out that our theta needs to be about 2.9 degrees, okay? So what this means, when we first shoot an X-ray, so when we shoot a crystal on the X-ray beam, we might catch the 140, the 42, 452 reflection on the detector. But then if you want to catch different ones like this one, we need to rotate our crystals, and then a different family of planes is now. So I got multiple lines here, just to show that there are different unit cells, all diffracting in the same direction, amplifying the signal, okay? So this is why we need to rotate the crystal while we're collecting data. So let's start to put some numbers. Let's start to put some names. Let's say this is carbon 1, carbon 2, carbon 6, sulfur, oxygen. So how do we know what we're going to amplify inside the unit cell? All we need to do the calculations for one unit cell. We know this is going to be amplified overall because the diffraction, the scattering in the phase of this atom is exactly the same as this, as this, as this. So let's just look at one unit cell. We can use an argon diagram, which is not very important. It contains an imaginary and a real axis. So we're going to use our C1. We can represent the amplitude like the length of the vector, and the phase as the angle with the real axis. So our C1, we know it's got phase 0, so it's lying flat. It will have an amplitude of about 6, which is the number. The amplitude is dependent on the number of electrons. The more electrons the atom contains, the bigger. Let's say C1 contains 6 electrons, so we get like a number 6. Now, if you look at our sulfur, it's like a quarter of the way between the two planes. So its phase will be around, let's say 90, 80 degrees. It will have a longer vector because it's got more electrons, so we'll have higher amplitude. If you look at oxygen 1, it's lying between the plane, so we know its phase is going to be around 180 degrees, so it's going to point the opposite direction. And we keep going this, so on and so forth, until we get our structure factor, which is our F. This is going to be our amplitude, this is going to be our phase, and this is going to be the family of plane that is related to this structure factor. So in this case, we can do the same thing. Yeah, our structure factor must mean like this, okay? So depending how the atoms are arranged within the planes of that family, you're going to have different combinations. So you might have a longer amplitude, so stronger amplitude, different phases. So for example, here we go our F2-110, which we know is going to be close to the detector because the D is very large. Here the D gets smaller, so they're going to go further. The higher resolution go further from the detector. But you see there are different intensity, okay? Because you can have a longer at low resolution, but you can have a shorter, a high resolution, or you could have a longer, a high resolution, and shorter, at low resolution, depending how they're arranged. Why did I not use F140 as an example on here? I just took two different examples because, as we know, 2-110 and 140 they cannot be found in the same image. Okay, because this one is diffracting on the detector in this orientation, the crystals, but this one would not, so it needs to be rotated. So I know that these two reflections will end up on two different images of detectors. Okay, but one more question is, why all the circular shapes in the spots? You see, they appear like circulars, okay? So this is to do with the fact that the detector, the diffraction happens in 3 dimensions, but the detector is just a projection onto 2 dimensions. But it's not easy mentally to see why we get these circles using real space crystal diffraction. There is another way to explain crystal diffraction. Instead of using the real space, it uses reciprocal space, which is much more, it cannot be pictured in that. You need to trust the maths and the geometrical construction, but it will give you an idea why you get all these circles. So I'm gonna go through it as well. So you remember you have your unit cells and your crystal lattice. Now we're gonna construct a reciprocal lattice by drawing a vector starting from the origin, which is going to go to a right angle against the family of plane. So this vector is going to represent the 110 family, and the length of the vector is going to be 1 over D. It's going to be the reciprocal space, okay? So which means the longer is this distance from the origin to the plane, the shorter this vector is going to be, okay? So these are the other family, 2-110, 120, 220. So the length of this vector is going to be 1 over D, reciprocal space, okay? Now, if you were to unite all the dots, you're going to get what's called the reciprocal lattice. You see 110, 210, 120, 220 here. So this now is your reciprocal lattice. How are we going to use this reciprocal lattice? We're going to pretend that this is our crystals. This is our HKL plane, dividing our 2 feet angle into 2. So it comes the X beam. Now, if we construct around the crystal a sphere of radius 1 over lambda against reciprocal space, and we're going to apply our reciprocal lattice with the origin here, now you will see that this point P lying on this sphere will be the distance between on and P will be the diameter, which is 1 over lambda sine phi of this angle, okay? But we know that OP, the distance between the origin and each of these points, is 1 over D, okay? This distance to this one is 1 over D in each family. So now if you rearrange this equation, this is nothing but our Bragg equation, okay? Which has been rearranged. We move the lambda to this side and D to this side. So which means that this sphere is called the Ewald sphere, okay? So it means that when we rotate in our crystal, instead of rotating the crystal in the reciprocal space, imagine you're rotating the reciprocal lattice around this point. So every time you rotate the crystals, different points are gonna come into projection onto the sphere. and only the points that are going to be on the sphere will be in diffraction, will be satisfying the Bragg equation. So now we can imagine that all these rays coming from this sphere, when you project them onto a 2D surface like the detector, they might start to look like these circles, which are called lunes, which is Latin word for moons, because they're like half moons. So now that we know our diffraction theory, how do we calculate our density, electron density? Which this is what we want in the end, no? So we can build our model. So we can decompose our electron density. Turns out that it can be decomposed into a series of summation. And guess what? The summation requires to know the knowledge of the structure factors. So we need to know all the amplitude, the phases, and the HKL numbers. So you remember, each of these is a structure factor, which will have an amplitude and a phase and HKL. So the more structure factor we can feed into this equation, the better our electron density is going to look like, okay? So if you only got this amount of information, we feed it into this equation. On the other side, we're going to get something look like this. But if we start to get high resolution, remember the high resolution away from the center of the detector, our reconstruction, our series is going to be more complete. Our reconstruction is going to look much sharper, okay? But how is this done? So imagine you collected a data set, let's say 360 degree, 1 degree each image. So you got like 360 images. First of all, you're going to find the software. You're using the software to find the spots, which can easily be identified by the software because they're much darker than the background. Once you find all the spots, you need to find the information about how far the detector is from your crystal, where exactly is the center of the beam, and the wavelength lambda you need to know. Once you have this information, using the Bragg law, you can reconstruct your reciprocal lattice. This is just a reconstruction. This is the actual reciprocal lattice. Okay, all these images have been aligned together. If you start to move them around, all these spots, do you see? You can start to see that there is. Now you see your reciprocal lattice. Okay, it is true. It does exist. Once you have this information, you can start to index them. You can start to say, okay, so this one is the 111, the 112, the 113. So that's called indexing. So now you can index all your spots. Once you index there, you can now once you have the reciprocal lattice, you can also calculate the reciprocal unit cell. So the reciprocal of the unit cell, the reciprocal lattice is the real unit cell. Okay. Then you can also know which Bragg lattice, which is the Bragg lattice. You can find how the crystal is oriented in the beam. Once you have all this information, then you can start to predict where the spots are. You can start to pick them automatically with the software. Of course, now you already have your HKLs. You can do the spot prediction. Once you find the spots, you can integrate them, which is going to give you the intensity. You have to do some scaling and merging. I'm not going into details, but you will get the intensity of each spot, of each structure factor, and the square root of the intensity is going to give you your amplitude. So now for each of these spots, you're going to know the HKL number and the amplitude. Now what you are missing is the phase. And this is known in crystallography as the phase problem. There are many tricks to get phases. I won't go into very details. I'll just tell you the name. One is called molecular replacement, which is becoming very popular today because you can use the AlphaFold models to estimate your phases, or you can actually experimentally calculate your phases. Something called a SAD method, another called the MAD method. But I won't go into details unless one day you will need them. Now, once we have the phases, we got all we need. Okay, we can start to reconstruct our electron density. Once we have our electron density, we can start to say, okay, this looks like a tryptophan. So we can start to build our model. Usually this is done with CUT. Once you have your model, you need to refine it, which means you need to fit chemically sensible models into the observed data, calculate the best electron density to criticize your model. This is usually done with software like RefMac, Phoenix, and Buster. And once you have your model, you need to validate it. You need to make sure that all the chemical bond and angle, everything makes sense. You didn't create some kind of monster. And to do all this, the software is all available on the LMB server. I look after the program, so they're almost up to date. If you find something is not up to date, just drop me an email. I will upgrade it. If you want to know more, I can suggest these books. These are like very good to understand the concepts. But if you want to know a little bit more, you want to go down to chemistry level, physics level, I can recommend these more detailed books. There are some lovely explanatory videos on the LMB page, done by most of these were done by people that actually wrote the programs to do data processing, like Andrew, Ari, Gary, Andrea. Me and Fabrice are always happy to help you if you need. Workshops. There are 2-day collection and processing training at Diamond. There is also a more intensive 7-day. This depends if your supervisor is happy for you to go for a whole week to Diamond, but I can recommend both. They're very good. So please, if you're interested, just let me know. I can book you for one of these. Now, more practical stuff. When to deploy X-ray now that we have a cryo-EM and AlphaFold. Okay. We know it was born in the LMB in the 50s, safety blossoming, 1890s. It was very good times. And then we start to push the sample side. It became very complicated. As you can see, earlier on, we had the bigger unit cells, which means we were trying to do bigger proteins, but the number of structure was diminishing. You can see the targets were getting bigger and bigger. So it was becoming very difficult to crystallize, which was becoming the bottleneck. Everything was there, the data collection, the data processing. The problem, people couldn't get the crystals, okay? There are some exceptional cases, like this huge virus was done by crystallography somehow. The TPAs here, the LMB, of course, the bank is ribosomes. These are all incredible achievements, but they all took like 10, 20 years. They were like life projects. Now, cryo-EM, we can do this much faster. In fact, the pressure is a bit off from crystallography now because cryo-EM is dealing with these difficult problems, with these large complexes. However, sometimes you can get regions in the EM where the resolution is not very good. So with this, we can just crystallize a small domain and do the crystallography and get very high resolution. At least this is what was done before AlphaFold. So these are some of the projects we did before AlphaFold. Like this was a low resolution filament. So what we did, we disrupt the filament formation. So we crystallized just the monomer. We could solve it to very high resolution and then plug it into the density. Same here, same concept here. This was done with limited proteolysis. As you can see, the resolution was very low. It's impossible to build the model. But then it was easy to plug the model into it. Once we solve it crystallography, high resolution. Similar here, similar concept. However, after AlphaFold arrived, most of the time AlphaFold is able to give you this model without being able to crystallize the domain. So if then the AlphaFold model is going to match your density, more or less, you know that you are on the right path. You can do some mutagenesis to check it. So we don't use crystallography for this very much anymore. Okay, these are some of the programs used to make the models, but of course AlphaFold is the best. Rosetta Fold is not too bad. Fire was, yeah, it wasn't very good. We used to use crystallography a lot when Fire was around. So when to use cryo-EM, also design the marks of your structures. I'll say if your protein is bigger than 200 kilodalton, definitely cryo-EM. If it's between 10 to 200, X-ray is feasible. I think you should first try cryo-EM. Below 40 to 100, probably X-ray is your best option. If you can get an AlphaFold model, X-ray should be very quickly to crystallize and solve it. As I said, if it cannot be crystallized, it's too small for EM. And even using mega bodies, nanobodies is not going to help. You can always try NMR, which will take 3 to 6 months, but you will guarantee your result. For small, very small protein. Membrane proteins, I suggest go to cryo-EM because they don't diffract very well membrane proteins usually. So AlphaFold 3, again, AlphaFold 3 is a technique. It's an empirical process, so you need to try. This goes is advantages, of course. You can work with multi-chromatic complexes. Non-protein molecules is a problem. Even AlphaFold, they said they can deal with metal things. It doesn't deal very well at all. Coil structure also seems to be a problem, so you might want to try to crystallize them. Small peptides, but very small also, that it doesn't work very well. You might need to crystallize them. Mutants, of course, it will always show the same conformation AlphaFold. Yeah, that's the problem. However, Sam is going to give a talk on AlphaFold, which I encourage you to go to, because he's going to have very good tips for you how to get the best out of it, because it is very useful. Like all techniques, it doesn't do everything, but it does many things. So now that there is AlphaFold, for crystallography, we got even less projects to do. For example, we work with RNA, which AlphaFold cannot deal with. For example, we work with phosphorylated peptides binding, which AlphaFold struggle with phosphorylation sites. We did the small molecules, which again AlphaFold cannot do. Drug discoveries, or we do synthetic amino acids. Again, AlphaFold cannot do synthetic amino acids, synthetic biology. Basically, that's it. Thank you for attention. If you have some questions, I'll be happy to answer. Thank you for coming.